Our next speaker is Michael Hölscher from University of Munich. Welcome, Michael.
Thank you very much, Carl, for inviting me and explain you why a university institution tries to develop a TB drug, and probably after the talk you know why. So BTZ is one of two novel class entities of the benzodiazepinones. I just want to tell you a little bit about history. Yesterday I learned how to do it, and now it's a new thing. So it's not a completely new compound. It has been discovered in 2008 by Vadim Makarov at the Hans Knoll Institute in Jena, so in Germany. And the patent rights lay with the Hans Knoll Institute in Jena. 2009, they outsourced the preclinical development to Alere, that was also located in Jena, and there were some pharma people from the eastern German part, and they took over the drug and preclinically developed it quite far. But in 2012, Alere decided to stop the drug development program and just focus on diagnostics. And at that time, about 2 years before 2010, Alere came to us within Germany and asked for advice on clinical development. And in 2012, the Hans Knoll Institute then came to us and asked if we would be interested to jointly work with them on the preclinical development. And this was a nice coincidence because at that time, the German Center for Infection Research, which I'm going to tell you a little bit more in a minute, was funded, and that was the purpose of the German Center, to bridge the gap between discovery and bringing products into practice. So the German Center for Infection Research decided to fund the finalization of the preclinical development program in 2014, and in 2015 we agreed between the four partners, including the Forschungszentrum, so the research center in Borstel, to do the drug development, and the project lead is with LMU. So just a few words on the German Center. It's founded in 2012. We receive an annual funding of roughly 40 million. It's 35 partner institutions, and you will see a map across Germany. It's a virtual center. It's a non-for-profit organization, and unfortunately the 40 million is not exclusively for TB. It's amongst the grand challenges of infectious medicine, and the primary focus is basically to serve as a catalyzer of the cooperating institutions, bring their preclinical and discovery expertise together and catalyze then the translation into products. We have 9 thematic units, and I'm going to explain this in a second, and 6 infrastructures, and is about 270 staff in funding. And it's all funded by the German ministry. And in some way we have to thank Mr. Obama, because he pushed our chancellor that Germany is doing very little in infectious diseases. So that's the map. It's 7 cities with 35 institutions, and one in TB, the 2 main partners are the Forschungszentrum Borstel and the LMU. And there's one specialty, because we have the regulators on board. So the Paul-Ehrlich Institute and the BfArM are members of the consortium, and we have some kind of extra official meetings with them, and they follow all our product developments very closely, give unofficial advice, and that helps us, being an academic, not to do too many things wrong. So that's the 9 thematic units. So you see TB is one of them. It bridges from emerging infections over HIV, over a unit called novel anti-infectives, malaria, gastrointestinal infections, and we have one unit that's a structural unit, which is called product development unit, and they help us a great deal. And for example, the clinical trial unit is directed by me. So the TB drug development platform is composing, so we also do compound search, and we the expertise is on the natural compound where there's a large library in Braunschweig at the HKI and also in Jena. Then we have the microbial, the search, the drug testing basically at Cologne with Jan Rybnicka, who was working with Stuart Cole before, and in Borstel. Then we do have the National Reference Center with Katharina Kranze, animal models, and then after that it moves to Munich with the preclinical development with my colleague Julia Dreisbach. And then if we ever manage to go into clinical phases, we will work within Panacea to develop those compounds. So BTZ is basically a DPRE1 inhibitor, which is necessary for the synthesis of the arabinofuranose, which is a cell wall component. And somehow this arabinoman haunts me over my life in TB research, because I started with LAM, say, like 10 years ago, and now I'm back with arabinoman. This mechanism is highly selective for mycobacterial species, and it binds covalently to the enzyme and blocks it irreversibly. So where are we in the drug development pathway? Still basically by Stuart Cole and Makarov, there was a lot of in vitro efficacy testing, and the MICs are between 1 and 10 nanograms per milliliters. All of the about 200 clinical isolates were tested sensitive. We are currently updating this library with the National Reference Laboratory, and the mutation rate has been assessed as 1 to the 8th. If we go to efficacy testing in mice, the old data show or didn't show, and that's why we went back and did additional experiments, didn't show a maximum dose effect, and I think that's what we have achieved here. You can see until 75 days with increasing dose, you have then here some kind of a saturation effect, and what you can see, it's much more efficient than INH at the current 25 milligram dose. And if you look to a different way of displaying it, you can see that we feel that between 250 milligrams and 1,000 milligram per kg, we have reached a maximum effective dose. There we have done in the mouse experiment some PK data, and we are still currently analyzing them. So in terms of safety pharmacology, there's no major signal. HERC is negative. There's no significant inhibition of enzymes and reporters. Irvin test is negative. Genotoxicity, all tests that we did were negative. Pharmacokinetics, no induction of any cytochrome assay that we did. And in vivo toxicology, we have completed rodent experiments with 7 and 28 days, and have established a NOAL of 170 milligrams per kg. And in mini pigs, that's somehow, I should have, so we have completed a 7-day, and the 180 milligrams is what we expect to be a NOAL. And if you would translate this into a human dose, it would amount to the enormously high dose of 36 gram for a 70 kilogram person, which we do not intend to give to people. And the currently anticipated dose of 10 milligrams per kg, we believe, and there is some historic data, that you reach at least 300 times the MIC in plasma. So that's where we are now. We are currently setting up with a company the GMP production and the drug formulation, and we hope to be in 12 months from today in human trials. So since we plan within Panacea, we just applied for funding in the future, and As you can see, if all goes well, we hope to be in phase 2A, probably end of 2019, and then move this through the step stage 2 design, as I explained yesterday. So that's basically what I wanted to tell you. Thank you very much.
Thank you, Michael. Any questions for clarification?
Good.
Okay, thank you. Our next speaker